Good morning, everyone. Let's get started. Welcome to our session, Airway Differentiation, Pathogenesis and Repair. I am Tayyab Rahman from University of Michigan. Also chairing this session with me is Finn Hawkins from University of Boston, Boston University. So we have an exciting session lined up today. We have 4 presenters. Unfortunately, one of our presenters had to cancel. So we're going to be a little flexible with time. We'll have 15 to 20 minutes for presentation. And after that, 5 to 10 minutes for question and answers. Hopefully we'll have good participation from the audience. You can come up to the microphone, ask your questions. You can also submit your questions online through using the mobile app. And with that, we'll get started with our first presenter, Feng Wan from UAB. And he's going to talk about pulmonary ionocyte-specific CFTR reactivation restores chloride absorption and secretion in CF.
Just a comment, it's really hard to hear the mic, so it's going to have to be pretty close to back here. It's hard to hear. Be close to the mic.
Okay. All right. Welcome, Feng.
Okay, thank you very much for the very kind introduction. So today I'm going to talk about how ionocyte-specific CFTR reactivation restores both chloride absorption and secretion in CF. So my name is Feng Wan. I'm an assistant professor from Division of Pulmonary and Cystic Fibrosis Research Center at UAB. Disclosure, there are nothing to disclose related to this presentation. So our group, as you know, our group used FARA as a model to study cystic fibrosis. And in this presentation, I'm going to talk about a couple of transgenic FARA models we used to address the debate over the pulmonary ionocyte function. So first, we use single-cell analysis to create a comprehensive single-cell map of the FARA airway epithelium, be able to analyze the gene expression across various cell types, including all the major cell types, such as secretory cells, basal cells, and synaptic cells, as well as the real cell types like neuroendocrine cells, ionocyte, and Topfer cells. Then look into the gene expression profile. So here is a gene expression a bubble plot. The darker color means the high gene expression. So we can find that the pulmonary ionocytes are highly enriched in many kinds of ion transporters, such as proton transporters, chloride transporters, sodium, potassium, and calcium. And not surprisingly, we found the cystic fibrosis gene CFTR is highly enriched in pulmonary ionocyte, along with many other chloride absorption and secretion transporters, such as CLCNK, BSND, which is involved in chloride absorption, and other chloride secretion protein genes like SLC12A2 and ATP1A1. Then next, we're trying to identify the ionocyte in the FARA trachea. So we performed immunofluorescence staining of the CFTR protein and the BSND, and we found a very good localization of the CFTR protein and the BSND in those pulmonary ionocyte, and the CFTR appear in a apical cap pattern in the ionocyte. And BSND is a subunit of CLCNK and CLCNKB, which is involved in salt absorption. And then subsequently, we moved to salt secretion ion transporters. So here we characterized the sodium potassium chloride co-transporter NKCC1. So we found NKCC1 is a good ionocyte marker, be able to enable ionocytes in a FARA trachea. And also we found a very good colocalization of NKCC1 and ionocyte marker ATP61G3. And in this image, they appear to be two population of ionocytes. One is the NKCC1 positive and ATP61G3 negative, and others are NKCC1 positive and ATP61G3 positive ionocytes. So, as cells NK BSND mediates chloride absorption and NKCC1 mediates chloride secretion, so those data really supports that ionocytes are capable of secrete and absorb chloride. Then next we generated the our liquid interface culture system. So this is a new culture system derived from ferret nasal basal cells, which can maintain very stable ionocyte numbers during the culture. As we can see in a wild type ferret culture system, it displays a good number of ionocytes in the culture system. And FOXI1 is a key transcription factor which is required for pulmonary ionocyte specification. And in the FOXI1 knockout airway epithelium, we found the absence of the BSND positive ionocytes, which is very consistent with ionocyte being consistent with FOXI1 being required for ionocyte specification. Then next we move to the CFTR staining at this in vitro system. We found those CFTR and BSND positive ionocyte, and the CFTR really shows the apical cap pattern, which is very similar to what we found from those in vivo samples on the ionocyte structure. So those really indicates, so this is a very good system to test ionocyte and CFTR function. So in 2023 in a Nature paper, we reported two different transgenic ferret lines. One is the FOXI1 CRE-ERT2, which is tamoxifen inducible CRE driver. So we inserted RS CRE-ERT2 into the 3' UTR of the FOXI1 gene. And the other transgenic ferret we made is a CRE responsive CFTR fluxed ferret. We inserted two NACFC site, which flux one of the CFTR exon 16. Then the double transgenic ferret FOXI1 CRE-ERT2 and CFTR flux upon tamoxifen induction can really make an ionocyte specific CFTR deletion. So this is a gene deletion at a cell type fashion. Then next we tested the CFTR deletion efficiency in a culture system. So we tested two groups here. One is the vehicle treated, which is a control group. We can clearly see the CFTR and the BSND colocalization. And in the tamoxifen treated group, it showed the absence of the CFTR protein expression as compared to the controls. Then next we move to Western blot to test the CFTR protein expression. We can find in the tamoxifen treated group, they showed the absence of the CFTR C-band as compared to the vehicle treated group. Now, after we set up this culture system, then we started to test the chloride absorption. So we tested three different groups here. One is the control, and the other is the tamoxifen treated, which is the ionocyte specific CFTR deletion conditional knockout group. And the other group we use a CF control cystic fibrosis airway epithelium. So the culture started from, so this is a Wilson chamber assay. We put the cultures into a Wilson chamber and started from a symmetrical high chloride, then moved to a chloride gradient with high apical and no basolateral chloride, which be able to drive a chloride absorption. So what we found here is that the control group showed a very large amount of chloride absorption. Then the ionocyte specific CFTR deletion group and the CF group, they show the absence of chloride absorption. Then by quantification, we can find that the phospholine and IBMX induced chloride absorption is significantly reduced as compared to the control, and the glytron-1 inhibitable chloride absorption is also significantly reduced. And we can find that the ionocyte specific CFTR deletion really phenocopies the CF group on the chloride absorption phenotype. So then we make a conclusion that pulmonary ionocyte mediate chloride absorption, and this is solely dependent on the CFTR protein. Then next we ask how does ionocyte contribute to chloride secretion. Then next we just play around the chloride gradient in the Wilson chamber system. We started from symmetrical no chloride and move into a no apical and high basal natural chloride, which be able to drive a chloride secretion. And similarly, we tested 3 groups here, control and ionocyte-specific CFTR deletion and CF. What we found here is that in the control group, there's a large amount of chloride secretion, and the ionocyte-specific CFTR deletion group showed a reduced chloride secretion phenotype. Then the CF group showed the absence of the chloride secretion. Then by quantification, we found that the ionocyte-specific CFTR deletion showed a significantly reduced the phosphocorneal and IBMX inducible chloride secretion and GLUT1 inhibitable chloride secretion. And the reduction is about 70% in the conditional knockout group. Then the residual chloride secretion from the ionocyte-specific CFTR deletion group, the cell, which cell type is contributing to the residual secretion remains unclear, which requires secretory cells and basal cells driver to figure out. Then pulmonary ionocyte partially regulate chloride secretion through CFTR. So next we ask, does reactivating the CFTR only in ionocyte restore both chloride absorption and secretion in the CF? So kind of to reintroduce the CFTR gene into the ionocyte in the context of cystic fibrosis and to test how the ion transport. Then so here I'm going to introduce a new CF model, so which is a CFTR EGFP is a conditional knocking model. So by which we insert a mini gene which is query-responsive with the lock-speed side and splicing acceptor EGFP on the stop side into a CFTR intron 1. So this ferret can be able to track all the CFTR expressing cells by EGFP, and the stop side are able to hijack the CFTR gene expression. Then upon expression, the mini gene can be accessed to make a CFTR reactivated in a CF. So then we did a immunofluorescence staining. We found out that the EGFP signal is really intense in the BSND positive ionocyte. Then next we kind of breed this CFTR EGFP ferret into the FOXL1 T2 and the CFTR G551D ferret. We have a triple transgenic FOXL1 T2 and CFTR EGFP and G551D ferret. So this CF ferret with one allele carrying CFTR EGFP and the other allele carrying the G551D, which can help us to help the CF ferrets to survive with the CF modulator VX770. Then next we tested the inducible CFTR reactivation. So we have 2 groups here. One is the control and tamoxifen. We found out the control group showed an intense CFTR, intense EGFP signal and the BSND co-localization in the ionocytes. In the tamoxifen treated group, when the mini gene is accessed, the EGFP signal is turned off. Then next we test whether the CFTR reactivation in ionocyte can restore both chloride absorption and secretion. And then we started to using a Wilson chamber to test chloride absorption. And similarly, we started from the symmetrical high chloride and moving to the asymmetrical with apical high chloride and basal natural low chloride to drive a chloride absorption. Then to test the chloride absorption. So we tested 2 groups here. One is the control group, which is vehicle treated, and the other is tamoxifen treated to be able to reintroduce the CFTR gene in ionocyte in CF. What we found here is that the CF group showed absence of chloride absorption, and when CFTR is reintroduced into the ionocyte in CF, we can find the restored chloride absorption. Then by quantification, we found that the phosphorylate IBMX induced chloride absorption is significantly increased, and the glytron-1 inhibitable chloride absorption is significantly increased as compared to the controls. The next we test chloride secretion, and we play around the chloride gradient. We started from a symmetrical no chloride and moving to a symmetrical apical low chloride and a basolateral high chloride to drive a chloride secretion. And what we found here is that the control group, control CF group showed the absence of phosphorylate and IBMX induced chloride secretion, but the tamoxifen treated CFTR reactivated ionocyte group showed a large amount of chloride secretion. Then by quantification, we can find that the CFTR chloride secretion is restored in the ionocyte-specific CFTR reactivated group. Then next, we're trying to address how the L surface liquid volume changes. So we make those the FOXL1 T2 CFTR conditional knocking basal cells, then let them grow in the ALL culture for 2 weeks. And the second week we put on tamoxifen, and then we use the micro OCT to measure the L surface liquid volume. And what we found here is that in the CF group, they showed very little L surface liquid volume, but the tamoxifen treated the CFTR reactivated ionocyte group, they showed a large amount of L surface liquid ionocyte. Then by quantification, we found that the L surface liquid volume is significantly increased in the ionocyte specific CFTR reactivated cultures. Then goes to my summary. So ionocyte specific CFTR facilitates both chloride absorption and secretion, and really supports the dual role of ionocyte in airway fluid homeostasis. And so whether this the upside rules are carried out by separate ionocyte subtypes or by the same ionocyte remains clear. So which is the one of the direction my own lab is going to investigate. Okay, and then comes to acknowledgement. So thanks my whole lab, my current trainees, Dr. Mei Hua Hao, Hank Chen, and Gabriel, and my former trainees, Adrian, Frank, and Grace Gesell. And a special thanks Dr. Zhang Engelhardt Lab. So this work is supported by Dr. Zhang Engelhardt and the whole Dr. Zhang Engelhardt team, Dr. Xin Yan, Xinsen, Edward Tang, and Xiaomin Liu, and Java Yalin, and all the Engelhardt Lab members. And also special thanks to the UAB CFRC, Dr. Brian Davis and Dr. Kao Fu, and my also special thanks to my collaborator Haber Lab at Harvard, Dr. Adam Haber, and the Federal Resource Center. And my research program is funded by the Cystic Fibrosis Research Institute and the Cystic Fibrosis Foundation and the UAB Division of Pulmonary Research and Development Funds. So that's all my presentation. I'll be happy to discuss any ionocyte function with you guys in the audience.
Hi, I'm Yi from the foundation. So it's great job, the wonderful results. So here is a question for you. All these cases, do you test non-gradient these conditions? How to the function data?
So at this point, we on the Wuxing chamber, we didn't test the symmetric long gradient. But the other assay we did is the organoid swelling, because the time limit I couldn't put in here. So in those organoid swelling assay and on the fluid secretion is more like symmetrical conditions. So we can find the ionocyte-specific CFTR deletion really inhibits the organoid swelling,
Okay.
which reflects the ionocyte play a role in fluid secretion.
Okay. The second question is you talk about the expression CFTR in the this variants. My question is, do you test, for example, with the blood, use this use wild-type variants? So is no expression CFTR or the high expression compare the wild-type variants? My question, if in some cases you overexpression CFTR in this CF, this case, what's happened for you?
Oh, you mean like overexpression?
Yeah,
CFTR
so maybe CFTR expression higher than wild-type variants.
Oh, I think we did both wild type and conditional knockout controls. Yeah. So we didn't find a huge difference.
Okay. For example, in the future working, do you have some plans for the cooldown optimization and overexpression CFTR higher than wild-type variants?
We haven't thought about that. Maybe in the future we should use those viral vectors to overexpress CFTR in those cultures.
Yeah, thanks.
To test.
Thank you so much for that. I had two observations. One was compared to what I've seen in humans, there seems to be many, many more ionocytes in ferrets. Do you think that's true? I mean, it looked like there was definitely more than 1% of the cells were ionocytes, whereas in human they seem much more rare.
Well, I think in the human data set, all the conclusion is coming from single cell sequencing. And for the single cell sequencing, they're really they have some limitations in capturing the ionocytes. So it's really difficult to tell what's the real percentage of ionocytes in the human samples. And I believe there's no study. Oh, there's one study from Dr. J. Gopal lab. They did a comparison between the proximal trachea to the distal airways. They didn't find a huge difference.
Well, I know that, but it just still seems like the number of ionocytes in ferrets seems really high compared to what we see in human. We probably should just try to resolve that as a community, I guess. Yeah.
Well, we didn't do this kind of comparison.
Yeah, I know. Maybe somebody else knows. Yeah. And then the other thing is, can you speak to the phenotype in the ferret with these various ionocyte models? Like, I know the CF knockout ferrets get pretty bad respiratory disease early, right? With infections and stuff. So can you speak to the changes in the lung phenotype that you see when you have correction or defects in CFTR in ionocytes?
Are you asking about the ionocyte-related ferret models? We're just asking about G5
so how do they compare to the CF knockout models in terms of mucus obstruction, bacterial infection, just the general airway disease that you see in a CF?
Okay. Yeah, I think I got you. So some of the models I introduced here, one is the FOXL1 knockout. So it's an ionocyte depletion. And speaking about the mucus obstruction, we didn't see the mucus obstruction in the FOXL1 knockout ferrets. There are a couple of reasons. One is the FOXL1 knockout ferrets are really difficult to rear, and the majority of them will die, and they cannot get into adulthood. So we didn't find the mucus obstruction phenotype in the FOXL1 knockout. And the other way is there's a selection bias on the FOXL1 knockouts. And for the ionocyte-specific CFTR gene deletion ferrets, I think all the gene expression, either inactivation or reactivation, is temporal. So it's not starting from the developmental stage. And we didn't evaluate the mucus obstruction in a similar manner as the CF ferrets. Yep.
Very elegant studies, congratulations. I was wondering whether you could speculate when secretion is done by CFTR in secretory cells versus ionocytes? Because you clearly showed that it's not all secretion that's mediated by ionocytes. So in a physiological picture, like in airways in vivo, do you think they secrete at the same time, just at different ratios, or is it differently stimulated?
Yeah, this is a really good question. It's about because there are two group cells are playing a secretion role. One's ionocytes, the other we're not sure. So I think it really requires the secretory cells specific CFTR deletion tools, which I believe some of the UNC group is working on that.
Okay,
So,
thank you.
Yeah.
Can you speculate as to whether there is just one ionocyte subtype that is mediating both secretion and absorption, or are you leaning towards whether there are two subtypes with distinct functions?
Yeah, some of the data we generated, because I couldn't get it here, show here. So it's about ionocyte subtypes, whether one subtype is working on the secretion part and the other is working on the absorption part. In some of my recent data, they showed a very distinct population of ionocytes, but we're still trying to figure out which subtype works, like the function. So that's the ongoing project we're trying to address. Yep.
May I ask one last question? Have you thought from a therapeutic perspective about the potency of an individual ionocyte and how far of a field effect a single ionocyte could have on ASL hydration, for example?
So can you repeat your?
I don't know if this one. The potency of an individual ionocyte, how the field effect of a single ionocyte is what I'm curious about. Have you thought about that?
No, I think some of the study we did is on how the ionocyte specific CFTR deletion, in a culture, they are able to induce basal cells to regenerate new ionocytes. So that's some of the part we're working on.
Okay.
Yeah.
Thank you very much.
Yep. Great. Thank you so much.
I'd like to welcome our next speaker to the podium. Stuart Rowlands is an attending pediatric pulmonologist at Children's Hospital, an instructor of medicine, and he's going to talk to us today about an iPSC model of ionocytes.
Thanks, Finn. Okay. Thank you for that introduction, Finn. So my name is Stuart Rollins. I'm a young pediatric pulmonologist working in Ruby Wong's lab at Boston Children's. And I'm going to be talking to you guys today about leveraging an induced pluripotent stem cell airway. Can you hear me now? All right. Well, I might lean in a little bit. Forgive me if I look awkward. Just look up at the slides. So as I was saying, I'm Stuart Rollins, young, 2 years out, pulmonary faculty at Boston Children's. I work in Ruby Wong's lab, working on induced pluripotent stem cell airway models of CF and other diseases. And I'm going to be talking today about leveraging that technique to study pulmonary ionocytes. The generic NACFC disclaimer. I'm not far enough in my career to have any relationships to disclose, but a man can dream. And I knew Feng was going to come in front of me, so I don't have a lot of background on ionocytes, but suffice to say it's been a few years now since single-cell RNA-seq at a few labs, including the Rajagopal lab, helped identify rare airway cell types, including pulmonary ionocytes, which I think a lot of us got interested in because they had such a high proportion of CFTR transcript. It should be noted they're not the highest number of cells that express CFTR. It's a relatively rare cell population, about 1% by single-cell RNA-seq data. And so there are a lot of different model systems that have been used to study ionocytes, from initial studies done in murine models to studies using human primary bronchoepithelial cells to really a tour de force of ferret work done by Feng and Dr. Englehart's lab that have helped to contribute sort of to our understanding of ionocytes and their function in airway physiology. And so our lab, working with Dr. Wong, we focus on using an induced pluripotent stem cell model. So for people who aren't familiar with iPSCs, these are induced stem cells. So you can take patient samples, either skin fibroblasts or serum sample, blood samples, and you can reprogram them to a stem cell-like state, and then through a directed differentiation protocol, give rise to different tissues. So in our case, we're deriving airway epithelium. I should say this is the one slide I will have going over a very complex process. This is summarizing well more than 5, 6, 7 years of Dr. Hawkins' life. My mentor, Dr. Wang, learned it from him and Dr. Daryl Cotton working at BU in the CREM, and then she took it back to Boston Children's, where I've been leveraging it. So essentially what we're doing is we're going from a stem cell and recapitulating developmental embryology through endoderm, anterior foregut, lung progenitor, so NKX 2.1 positive cells, to a mixed airway organoid model. From there, you purify out a population of basal cells, and then those you can treat almost like HBECs. You can plate them on ALI cultures, and then you can get a pseudostratified airway epithelium that we're used to working with. Now, the advantage of an iPSC system is you can do some rather complex gene editing on these stages at the iPSC stage when they're a clonal population of cells. So being interested in CF, pediatric airways disease, and iPSC airway models, we thought, how can we leverage this technology and this model system to help further study ionocytes? And so what we first did was we took a healthy donor, reprogrammed, these were based on RBCs, to an iPSC line, and then that underwent CRISPR-Cas9 gene editing to knock out FOXI1. And so at this point, you have a syngenetic pair. There's the same genetic background from a healthy individual. The only difference is we remove FOXI1 expression. Subsequently, both of those lines undergo a parallel directed differentiation based on the protocol I just summarized on the prior slide. One of the first things we were nervous about when we started this study was, well, if I take out a transcription factor, how's that going to affect directed differentiation from a stem cell state? Thankfully, both lines were able to make a good quality ALI culture, and I'll show you some further data on subsequent slides. But two of the markers that we look at here, one is CPM, which is a proxy for NKX 2.1. We also do quality mark staining for NKX 2.1. It's just, that's an internal transcription factor. So once you're confirming that, the cells have to be fixed and stained so you can't progress. So we use CPM as a surface marker. And then for basal cell purification, we use NGFR. The cells undergo multiple NGFR sorts to eventually get to a high purity of NGFR positive cells for basal cells. This is a modification to the original protocol that Dr. Wang originally published a few years ago that showed successful differentiation of ionocytes from iPSCs. This isn't the exact timeline of how we designed these cultures, but subsequent to the experiments that we did with these two lines, we realized it would be very useful if we could turn FOXI1 expression back on. Based on what we were seeing in that knockout line. So we took that iPSC FOXI1 knockout line, we had it undergo a second round of CRISPR-Cas9 based gene editing in order to insert a doxycycline-inducible FOXI1 with a co-expressed M-cherry reporter. So this gives us a total of 3 lines, a wild type line, a line lacking FOXI1, and a line lacking FOXI1 where we can conditionally turn FOXI1 back on with a fluorescent reporter to track that. These are all within the same genetic background. And that third line also had acceptable CPM, NGFR sorts, and was able to make it to air-liquid interface cultures. So our first round of characterization was basically just to see when we knock out FOXI1, do we see a reduction in FOXI1 expression, which we do at both a protein level and a transcript level. We saw a significant reduction, but not a complete elimination of CFTR, as well as significant reduction in the ionocyte markers ASCL3 and BSND. There's just some representative microscopy of 2-week-old cultures. Here is further characterization of our wild type and our knockout syngeneic lines. I will say even before we did this staining and the subsequent transcript analysis, one of the things you notice with these cultures is they do have thicker, more tenacious mucus. You find yourself needing to incubate them a little bit longer with either warm PBS or Krebs buffer to help clear off the apical mucus in the transwell cultures. And sure enough, when we looked at these cells, we found that our FOXI1 knockout actually did have an increased proportion of secretory cells, stained for by MUC5B here, you'll see in the purple. We also noticed both lines did have a presence of alpha-tubulin, although interestingly enough, our knockout culture seemed to have a decreased amount of ciliated cells relative to the secretory cell population. And then of particular interest, actually in light of a paper recently published by Jay Rajagopal's group with Viral Shah, we also saw a significant increase in tough cells, PNEC markers in our FOXI1 knockout culture, so an increase in ASCL1 and POU2F3. And that may have something to do with they're proposing a common tough ionocyte progenitor cell. And so it may be something where if you're knocking out the ability of a cell to become an ionocyte or a FOXI1 positive cell, you may be shifting some of those cells over towards a different cell type. Now, I will say that's a little speculative on my part, but I think it's something that's worth further exploration. I'm sure Jay's lab is doing it. So having done these basic characterizations, the thing we want to know is, okay, if we knock out FOXI1, what effect, if any, does that have on CFTR-mediated ion transport? So we did Using's chamber studies. I will say these are symmetric chloride buffer conditions with our wild type and our knockout line. So our wild type line, we did Using's chamber analysis. We saw a good, decent ENAC response, good forskolin response to CFTR inhibitor 172. And when our knockout cultures, we saw a significant reduction in our forskolin and CFTR inhibitor 172 response, though not a complete elimination. And so presented here is the average of 4 paired experiments, as well as representative microscopy of the wild type and the knockout cultures with BSND as a staining marker of mature ionocytes. That stain isn't from the same wells that underwent Using's, it's from paired wells harvested on the same day of the Using's trace. So having seen that phenotype, that's when we went back and we said, oh, so if we're knocking out FOXI1, we're seeing this EPHIS data. Well, what would happen if we could restore FOXI1 expression within those knockout cultures? So that's when we went back and we had our second pair of lines with a FOXI1 inducible line. So this is now all in a FOXI1 knockout background. First thing, just to characterize, okay, in our knockout line, when we add doxycycline to trigger an expression of FOXI1 to induce some ionocyte formation, does that result in restoration of ionocyte markers? Sure enough, it does. We see restoration of BSND and FOXI1, an increase in CFTR, although it's notable, again, the FOXI1 knockout cultures weren't without CFTR expression. And so going back to repeat Using's data, we were able to show that when we treated our FOXI1 overexpression line, which again is in a knockout background, so it doesn't have any baseline FOXI1 expression or ionocyte production, when we restored the presence of those ionocytes, we were able to see a restoration in forskolin and CFTR inhibitor 172 response. And being CF clinicians and physicians, one of the things we're interested in is, okay, so this is sort of interesting, but what would happen if we took this FOXI1 overexpression line expressing wild type CFTR and we introduced that to a CF airway epithelium? And so we saw a very basic sort of prelim experiment taking an iPSC line derived from a patient with Delta 508 homozygous genetics. We mixed that with a smaller proportion, about 5% of our doxycycline inducible FOXI1 M cherry line with wild type CFTR, created a mixed culture, and then either treated paired cultures with a vehicle control or doxycycline to trigger the emergence of wild type CFTR ionocytes. And sure enough, what we found was within our Delta 508 homozygous line, without any triggered FOXI1 differentiation, we didn't see much of any forskolin or CFTR inhibitor 172 response. But when we triggered the emergence of wild type CFTR ionocytes through FOXI1 overexpression, we were able to restore that function, both in terms of a forskolin response and a CFTR inhibitor 172 response. So to summarize sort of what we showed with our FOXI1 related lines, we showed that FOXI1 knockout decreases ionocyte formation and CFTR-mediated ion transport. We saw a decreased expression in ciliated cells and an increased expression of secretory cells within our knockout line. Restoration of FOXI1 expression within the FOXI1 knockout line background increased CFTR-mediated ion transport, and the addition of wild-type CFTR ionocytes within a Delta 508 homozygous CF background appeared to rescue CFTR-mediated ion transport in our iPSC airway system. And so with a little bit more time, I want to draw some attention, and really within the past year, there's been a lot of really good work from a bunch of different labs looking at the role of ionocytes and what I would characterize as perturbed airway epithelium. So airway epithelium exposed to relevant inflammatory conditions, cytokines, airway epithelium derived from non-CF disease, particularly asthmatics. So I would draw your attention actually to a great JCI paper out of David Stoltz's lab, where they looked at prolonged IL-13 exposure and actually showed a very dramatic shift in the proportion of ionocytes to secretory cells, as well as an interesting emergence of CFTR negative BSND positive ionocytes. There's a lot of data. I've highlighted one with people actually looking at primary data from cultured HBECs or respiratory nasal cultures of asthmatics, where people have shown a decrease in CFTR function as well as FOXI1 positive cells, particularly within both eosinophilic and non-eosinophilic asthmatics. It's not listed here. I probably should have had it, but Jay Rajagopal's group, Viral Shah's the first author, had a great Nature Communications paper looking at the effect of type 2 cytokines and IL-17 on the relative expression of ionocytes and pulmonary neuroendocrine cells as well. That came out a few months ago. And then lastly, and I promise this, I'll get to how this is relevant, but of particular interest to some of the other work we do, there's actually a large cohort study published in the Blue Journal that was really more of an asthma genetics paper that was looking at different putative regulatory SNPs. So this is almost 300 kids where they did primary nasal cultures. And if you dig through that supplementary data, you'll actually find that in the asthmatic children compared to healthy controls, there was a significant decrease in FOXI1 and ASL3 expression compared to healthy. And then even within comparing severe asthmatics to mild asthmatics, an even more significant reduction in FOXI1 and ASL3 expression. I mention this because in addition to our CF work, we've also leveraged this iPSC-based system to model genotype-specific disease responses. So we have several asthmatic patients that we've done iPSC reprogramming on, where we're interested in the effect of small genetic changes, putative regulatory SNPs, in affecting their response. And so we recently had a little letter published in the Blue Journal where we had taken 3 asthmatic patients with different putative asthma risk alleles and characterized their response to IL-13 and poly-IC. And we didn't get into it in that paper in terms of ionocyte data, but suffice to say we were able to show that in our high-risk asthmatic allele group a stronger response to IL-13 in terms of secretory cell formation, as well as an increased inflammatory response to poly-IC, which is like a viral mimetic. And then with David Stoltz's data and the data coming out of the Rajagopal lab, I actually went back and analyzed some of those samples. And what's interesting is in our high-risk asthmatics, iPSC-derived cultures, we were seeing actually a significant production of BSND and ASCL3 transcript. It's not significant, but sort of trending that way in terms of CFTR expression. And this is all obviously extremely prelim, but I think when I think about ionocytes and secretory cells and the airway epithelium and how this story is advancing, I think the next things that people are going to want to look at is what are the effects under perturbed conditions? What are the effects under infectious conditions or long-term inflammatory stimulation? It's not just the baseline physiology. There's so much good work that's being done in terms of characterizing ionocytes, the different subtypes. And I think the next step is to think, well, what does it look like in our patients, right? Whether that's our patients with CF and chronic inflammation, I'm a pediatric pulmonologist, so my patients with asthma, everyone in between. So I just thought it was a little interesting. So that's what I have for today. None of this is possible without a lot of people. I'm a MD by training. Really, I've been with Ruby now for 4.5 years. And she had worked with Finn and Daryl beforehand. I wouldn't be a researcher without the Cystic Fibrosis Foundation. They got me the grants to protect my time in the back half of fellowship and the beginning of my training. They're still funding me now somehow. And it's always a pleasure to be at this conference and talk with people about different things and what's going on. I think it's fantastic. I'm happy to talk about any of this work. The lab itself has a large bank of iPSCs from both CF patients and non-CF patients. And I think it's a powerful tool in terms of studying both mechanistic effects as well as providing a human-based model system, particularly as we're seeing some shifts in NIH funding to promote human-based systems. But the truth is, you still are going to need murine models, ferret models, high-fidelity models. There's only so much you can do in a dish. With that, I'm happy to take any questions.
Nice talk. I like it. One technical question. So when do you restore the FOXI1 expression? When did you do that? So like before ALI culture or a week, 1 week through, 2 week through?
We do it 4 to 5 days post plating at ALI culture. So starting with like a pure population of basal cells, it works out to being about 48 hours post airlift. If that makes sense, if you're familiar with how we do these cultures.
Yeah, it does. So one question. So have you tried to do that like later on?
So it's something we're absolutely interested in doing to track late coming emergence. I didn't mention this, but one of the other sets of tools we're designing is a reporter system for both FOXI1 and BSND with a triggerable caspase-9. So essentially a triggerable deletion of mature cell types. So we're hoping to attack it from both ends, sort of triggering FOXI1 maybe at a mature airway state, and then also being able to trigger apoptosis of FOXI1 expressing cells in that same state.
Oh, great. So when you restore FOXI1, have you tried to measure how much FOXI1 do you express compared to the wild type?
So for the conditional overexpression, right now it's just transcript data, and by transcript it's an overwhelming amount more because it's a doxycycline inducible cassette. It's going to do a lot more. We're working on characterizing the actual protein level, as well as the proportion of FOXI1 positive cells, because there's not a uniform, depending on the amount of doxycycline you'll get sort of a variable response from this cell culture. So the safest way we found to do it is to just use low proportions of the FOXO inducible cell line and relatively high proportions of doxycycline to try and force all of them to be positive.
All right, thank you.
Now we gotta get back low. Great talk. So under goblet cell hyperplasia, right, in some of those images you could see that you just get like bonkers amount of CFTR in the goblet cells, right? So what are your thoughts on restoring CFTR function in an ionocyte that doesn't have CFTR?
So, restoring CFTR function in an ionocyte that doesn't have CFTR. So it's an interesting question. I think that gets down to the interesting work that is out of David Stolz's lab, where there may be physiologic conditions where you have CFTR-less ionocytes or BSND positive cells that aren't expressing CFTR, and then it raises a question of if you're triggering expression of CFTR within those cells, is that physiologic, or are you sort of are you doing something not physiologic that may produce a change on an Using's trace but isn't necessarily clinically relevant? I think it's interesting. I think there's a lot of different data that's come out, and my suspicion is that there's some degree of complex interplay between the shifts in ionocyte proportion, goblet or secretory cell emergence, and that we're going to find that it's really in our model systems, it may be very media dependent. My suspicion is in vitro, it's going to be dependent on the inflammatory conditions or what the actual airway's been exposed to. Like, I don't think it's going to be a clear answer that one cell type is more important than the other. I think it's going to be one of, this is a cell population that if it's not communicating directly with each other, it's certainly being influenced by the environmental conditions.
Mm-hmm. So do you think it's a pathological thing that they lost CFTR, or do you think that's a physiological response to goblet cell hyperplasia?
So I don't know.
I think
it's an important question. I guess is what I would say. I don't want to, off the cuff try and say it, but I think it's a very relevant question.
Yeah. Thanks.
Okay. Maybe too short for this. Nice work, Stuart. My question is similar to the first question. It's very interesting for me to see that at the end of your differentiation in your knockout, you have different cellular compositions, right? You have changes in secretory cells, ciliated cells, pulmonary endocrine cells, tough cells. Have you looking at the conditional restoration, the knock-in, the last experiment, of FOXI1 at earlier stages of your differentiation to see if that's impacting how these lineages are being directed.
So one of the things we're working on right now actually is to actually do a sort of more detailed time course based experiment where we're triggering it at different times and following one culture over a longer time point and just doing single cell across it to get a good sense of the transcript, and then in parallel taking cultures for protein characterization by immunofluorescence. Because I think one of the things that gets very tricky when you're talking about using an iPSC-based system where we're sort of relying on trying to recapitulate the development in primary cells, I do think we need to be extra vigilant about any conclusions we make about how the developmental pathways of the cells and what triggers them becoming ionocytes or different cell populations shifting. And so what I'm hoping to do is to capture sort of short intervals with both transcriptomic and protein-based staining to better characterize that. But I think it's an important question.
Hi, really beautiful system. So I wanted to ask a question about your cell mixing experiment. And I wanted to revisit Finn's question to the prior speaker about like the power of, or like the might of that single ionocyte and its kind of local environment. So are you guys planning to sort of really expand that and maybe go down to like, because I think you were doing like maybe 5%.
I have done it down to 1% and as high as 50. 50, everything will die when you trigger it up. If it's 50% and you use a high amount of dox and you push everything towards an ionocyte, the airway epithelium, your tear goes to trash. And down at 1%, we still get a pretty good Using's response, but I haven't done detailed characterization in quadruplicate to where I wanted to present the data.
And then have you been benchmarking the CFTR activity compared to wild type to kind of see like how much of that rescue that you're getting close to wild type?
They'd be compared to the not the non-edited, but the non-dox activated control. And so I think to your point, it would probably be ideal to set that experiment up. Basically have two controls: one, the mixed population that isn't triggered, and two, just a baseline population.
Awesome. Yeah, super cool.
Okay. Anything else? All right.
Thank you, Stuart.
Thanks, guys.
Okay. Our next presenter is Michaela Murphy from University of Colorado. And she's going to talk to us about inflammation, IL-1 beta, and its effect on ciliated cell fate, ciliogenesis.
Thank you. Thank you for that wonderful introduction, and I want to thank the speakers or the organizers for allowing me to come here and share my research today. We are squarely jumping away from ionocytes, and we are going into ciliated cells today on my talk. So before we get into that, I want to get into the lens under which I study ciliated cells in the context of cystic fibrosis. So normal airway epithelial structure and function is essential for healthy lung function. And in cystic fibrosis, we know the underlying CFTR dysfunction leads to a dehydrated airway surface liquid layer, and this leads to viscous mucus that is adherent to the epithelium. And in this process, we have something called the vicious cycle in cystic fibrosis. We have a buildup of mucus, we have inflammation, overwhelming intense inflammation, as well as infection. And this is a cycle, so it feeds back into each other. So I want to direct your attention here to some scanning EM images we have. First, from a healthy airway, you can see here that this healthy airway kind of looks like a shag carpet. The way that I describe it, it kind of looks like a very ciliated epithelium that kind of looks like this fluffy plush carpet. And you can see here that there's some mucus transport going on. This mucus is being put away by the beating of the cilia. In these areas from a person with cystic fibrosis who is an F508del homozygous, these are from areas of increasing apparent inflammation. And so area 1, you can see that this is a pretty relatively normal looking healthy epithelium. You can see the plush carpet of cilia here, as well as some of the mucus being transported. However, as we move along and we see this increasing apparent inflammation, we see some changes happening to this epithelium. We see a decrease in ciliated cells and this increase in goblet cells and mucus being found in these areas. So this is what we are going to term airway epithelial remodeling for this talk. So these are detrimental structural and functional changes to the airway epithelium. In cystic fibrosis, we see more mucus secretory cells as well as squamous cells and fewer ciliated cells. This does underlie disease symptoms, drives progression, and we do believe that this is focal, so not the entire airway is being remodeled at once, but episodic, and we do believe that this is variable in extent and possibly irreversible. So here's a schematic of a healthy airway. We have mucociliary clearance going on with this thin mucus. We have a selective barrier that's being maintained and robust generation of all cell types. However, in cystic fibrosis, we have this cycle that I talked about with intense inflammation, infection, and injury constantly assaulting this epithelium. Our questions in this are, what are the mechanisms of remodeling? So what is driving this remodeling phenomenon that we're seeing where we have this loss of ciliated cells and this overabundance of mucus secretory cells? And how can we get back to this healthy structure and function epithelial? And now we're going to dive into the ciliated cells. So ciliated cells, if you're unfamiliar with them, they are vital to CF host defense, as well as in a healthy individual. They really work. They're really the drivers of mucociliary clearance. They're also responsible for immune surveillance and response in the airway. Not only them, but they are a part of that immune surveillance and response to pathogen challenge. And there are receptors in cilia themselves that are actually direct targets for many pathogens, including many viral upper respiratory infections.
Maybe try just slightly back from the mic. We're getting...
Sorry. Ciliated cell dysfunction, we do know it is happening in cystic fibrosis, but it is underappreciated in CF. So these are transmission electron microscopy images from a healthy individual, and then as well from a person with cystic fibrosis that is homozygous for F508del. And you can see here in the healthy individual, we have the structurally intact axonemes, as well as they look like, nice hair-like projections from the cell. However, in a person with cystic fibrosis, we can see that we have compound cilia and structurally defective cilia. So we don't believe that CFTR dysfunction itself is causing the dysfunction that we see in cilia, but we think that it's acquired in cystic fibrosis. And when we say ciliated cell dysfunction, we are really talking about fewer ciliated cells with defective cilia, structurally dysfunctional cilia. We see decreased ciliary beat frequency and abnormal beat pattern, and this has been observed secondary in the literature, and we do believe, like I said, that this is secondary to CFTR dysfunction. We also have found from single cell data from our lab in CF plus ETI, which is a CF modulator, versus healthy airways, that these ciliary dysfunction does persist despite the addition of CFTR modulators. So in this graph here, I'd like to direct your attention to the left side of the panel. We see that there are more immature ciliated cells in CF, even with ETI, and that these ciliated cells that are present are really they exhibit this inflammatory programming. Alternatively, here we see that when inflammatory genes are high, we have a decrease in ciliated gene expression. So they're inversely correlated. So we've talked a lot about inflammation in ciliated cells and cystic fibrosis, but specifically, there are many pro-inflammatory mediators in CF, but we really wanted to focus on IL-1 beta. So we know from the literature, it's been documented extensively that IL-1 beta is the dominant pro-inflammatory cytokine in the CF airways. It's been shown to be a very big driver of epithelial remodeling, specifically in mucus cell metaplasia. Many studies have found this, and we have also recently prepared this manuscript is in preparation, but we can see here that IL-1 beta, this single cell sequencing in healthy individuals, shows that there's still a higher amount of IL-1 beta across the cell types, even with the addition of ETI in people with cystic fibrosis. So it does remain high despite this CFTR modulator therapy. And so for us, and when I came into this project, I wanted to ask, does IL-1 beta also directly impair ciliated cell formation? Does it impair ciliated cell fate? Does it impair ciliogenesis, cilia function as well? So before I get into the studies, I want to kind of walk you through the birth of a ciliated cell and how a ciliated cell is made. So we have this progenitor cell, and we move forward through basal body formation to the ciliating cell, where the basal bodies are trafficking to this apical surface. And then we have axoneme formation, where the axonemes actually extend from the basal body, and that's when you would see cilia. So there are kind of two different parts of this. So ciliated cell fate, so developmental pathways like Notch and Wnt signaling, as well as ciliogenesis, where ciliated cell specific transcription factors are turned on, and they impact all the steps of ciliogenesis. So you have some early ones, MCDIS and FOXN4, that are involved in like basal body formation, trafficking to and docking to the apical surface, and then FOXJ1, where we are seeing these axoneme formations. So now that we know how a ciliated cell is made, I want to introduce to you how I planned on studying the ciliated cell in this airway epithelium. So we're really fortunate that we partner with lung tissue donors, and we receive lung tissue post-transplant. And for me specifically, I work with lung tissue, so bronchi. I get the airways, I isolate the basal stem cells, expand them, and then I'm able to differentiate them at air-liquid interface into different cell types. And this is what this process looks like. So in early ALI, early cultures, we do submerged cultures where we're really just telling the cells to proliferate and grow. And then we move to an air-liquid interface setup where we are telling the cells to differentiate into different cell types. So the ciliated cells and mucus secretory cells. And this can this starts at ALI 0 and can go on till ALI 21, where you'll have a mature differentiated epithelium, and you can continue this culture for a while. So for the data that I'm going to be presenting to you today, and the studies that you guys are going to be learning about, we did two different time points for IL-1 beta exposure. We did an acute time point where we treated with IL-1 beta while the cells were growing at air-liquid interface from ALI 3 till ALI 4. This is a very early differentiation time point. We really wanted to hone in on a mechanism at this early time point. And then we also treated at a chronic time point where we went from ALI 3 to ALI 15, where the epithelium is mostly mature at this time, and we wanted to understand what chronic exposure to IL-1 beta during this differentiation and maturity would lead to. All of the donors that I use in the study are from healthy, all of the cultures in the study are from healthy donors. We haven't moved into the CF cells yet. And for the endpoints, we wanted to look at immunofluorescence for protein, and as well as functional assays for cilia function, and transcriptomic data to kind of help us pin down a mechanism. So jumping into the data, we found from some of the sequencing data as well as some of the immunofluorescence data that we were getting an activation of NF-kappa B signaling. So upon binding of IL-1 beta to the IL-1 beta receptor in cells, there are kind of a couple different flavors of response pathways that you can get. You can get the Phos-Jun AP1 complex response or an NF-kappa B signaling response. And we got a big induction of genes associated with NF-kappa B signaling. And so that to us indicated that we had some NF-kappa B signaling activation here. Very much what we expected to see. We saw that we expected to see mucous cell metaplasia being induced by this IL-1 beta. Here you can see a control treatment. So there are, in fact, mucus cells present in the epithelium. But when we add the IL-1 beta to that chronic time point, we see a huge induction of mucus cell metaplasia. And this is just data from one donor, this is just IF from one donor, but we calculated this for all 6 of the donors that we used for the study, and we do indeed see a big induction of MUC5AC positive and MUC5B positive cells following chronic IL-1 beta exposure, which is, as I said, very much expected. We also went on to determine the impact of IL-1 beta on ciliary function. So we first wanted to look at mucociliary transport. So mucociliary transport is a metric that is influenced by both mucus, the viscosity of the mucus, as well as the cilia, the function of the cilia, the beating of the cilia. And we find that the mucociliary transport is diminished. So fluorescently tracked beads, we can see the control. These beads are being moved by the cilia. They're being pushed. They track for long distances, and it's really a coordinated movement in one direction. However, when we exposed these cells to chronic IL-1 beta, we see that we kind of have a arrest of the mucociliary transport. Many of the beads are not moving at all. If they do move, it's a very short movement, and it's not really a coordinated beat in one direction. To go along with this, we calculated ciliary beat frequency, and we found that the ciliary beat frequency is reduced with chronic IL-1 beta treatment. So now the star show of my presentation and what we study in the lab, ciliogenesis and how IL-1 beta is impacting ciliated cells in a ciliated cell fate manner, as well as ciliogenesis. So before we move into that data, I just want to draw your attention back to the ciliated cell fate and ciliogenesis pathway. So we look at this under a couple of different lenses. So first, ciliated cell fate and the developmental pathways that signal a cell to take the ciliated cell lineage, and then the ciliogenesis factor, where the cilia are actually being, the basal bodies are actually moving to the apical surface, the axonemes are extending, and the cilia are being formed. And what we find is that with chronic IL-1 beta treatment, we see fewer ciliated cells and more immature ciliated cells are made. So this first panel that you see here are quantifying basal bodies and axonemes, and in the control you can see that we have a good balance of cells that have both basal bodies and axonemes. However, with chronic IL-1 beta treatment, we lose the colocalization of the basal bodies and the axonemes, meaning that some of the basal bodies that are present do not have axonemes growing out of them, which means that these cells are immature. And this is a little graphic here to show that these are just being retained in the cell and they're not docking properly and not extending axonemes from them. When we look at transcriptional factors of ciliogenesis, we can see in the control that we have a lot of FOXJ1, which is a marker of late marker of ciliated cells, and we have a decrease in FOXN4, which is an earlier ciliogenesis marker. However, with chronic IL-1 beta treatment, we see the exact opposite trend. We see less FOXJ1 positive cells and more FOXN4 positive cells, indicating again to us that we're really not getting the full, these cells are not becoming fully mature ciliated cells. We also quantified this with FOXJ1 positive cells. We see a reduction in chronic IL-1 beta treatment, and we see an increase in the fraction of immature ciliated cells, which are these ODF2 positive cells and acetylated alpha tubulin, which is an axonemal marker that we see here in red, and we see that there is an increase in these immature ciliated cells. When we look at the transcriptomics data, we got a lot of genes back associated with different parts of development of a ciliated cell. We have genes associated with the axoneme motility, intrafragillar transport, basal bodies, transcription factors of ciliogenesis, and early ciliogenesis markers. And what we found was that the acute time point, we get this big suppression or downregulation of transcription factors associated with ciliogenesis. In the chronic time point, we see this suppression of later ciliogenesis regulators, so not being able to fully form a mature ciliated cell, but we do see this induction of early ciliogenesis regulators, so they are trying to commit to the fate but are unable to reach full maturity of a ciliated cell. So the chronic IL-1 beta is blocking this transition in ciliogenesis. So by this point, we have shown that IL-1 beta does lead to this epithelial remodeling that we're seeing in cystic fibrosis. And when I mentioned in the beginning of the talk, we're really also focused on how do we get back here to a healthy airway epithelium structurally and functionally. And one of the things that we wanted to propose is inhibiting the notch signaling or the notch pathway as a mechanism of preventing ciliated cell loss due to exposure to chronic IL-1 beta. And we found that this notch inhibitor co-treatment, so treatment of notch inhibition or of a notch inhibitor while we also treated with IL-1 beta, was able to restore the ciliated cell numbers. So in this data panel here, FOXJ1 is shown in red. The first panel is the control, and the second panel is the chronic IL-1 beta treated. With untreated, we see that exactly what we expect to see, a large number of ciliated cells. However, in the chronic IL-1 beta treated, we see this reduction in FOXJ1 positive cells, or this decrease in ciliated cell number, which is very characteristic of the things that we've seen so far. However, when we add DAPT, which is the notch inhibitor that we use, we see there's no difference at the control level with the amount of ciliated cells. But when we add this co-treatment in with the chronic IL-1 beta treatment, we see that we're able to increase ciliated cell number. And so we also quantified this with the amount of FOXJ1 positive cells in the area. And we see that with IL-1 beta treatment, we are able to rescue the ciliated cell loss with DAPT treatment. However, it's still not up to the level of the control or the non-IL-1 beta treated cells. And so all of my work today, the conclusions that we gained from this is that ciliated cell dysfunction does exist in CF. I hope I've convinced you of that today, and that unfortunately this does persist despite CFTR modulator therapy, and that these ciliated cell defects, the decrease in ciliated cells, as well as defective ciliogenesis and defective ciliary function, contribute to these mucociliary clearance defects that we see, and that this remains an underappreciated area of study in CF. We have shown that IL-1 beta does cause ciliated cell dysfunction, and we see fewer ciliated cells, as well as defective ciliogenesis and defective ciliary function, and that directly targeting the ciliogenic pathways could improve remodeling and mucociliary clearance in CF, and we have shown preliminary data with notch inhibition blocking the deleterious effects of IL-1 beta that we observed. With that, I would like to acknowledge my lab. Sonia is the tech that's been there with me the whole time. She's been invaluable to this. Sierra did a lot of or pretty much the bulk of the transcriptomics. Chris Evans' lab, as well as Katie Hazard's lab, they've been invaluable guidance to me during this time. And I would also like to take this time to thank the lung tissue donors for their selfless act to push science forward after their death. So thank you, and with that, I will take any questions.
Thank you. It was a fantastic talk. Just 2 points. I know this is all in wild type cultures, not yet CF, right?
Yes, I have not went into CF cultures yet.
Because one of the things I was wondering if you'd thought about, because it's interesting that you can, affect notch and maybe ameliorate some of your IL-1 beta phenotype, but clinically we have drugs that target IL-1 beta specifically. Like we'll use an ILD, like canakinumab targets just IL-1 beta. It's used in some interstitial lung diseases. And so if you're hypothesizing that there may be an IL-1 beta mediated effect of like long-term inflammation in CF affecting your cilia formation, I'm wondering if you would think about, when you move towards those diseased cultures, if there might be some utility in looking at already approved drugs that might directly affect that inflammation.
Yeah, absolutely. That is a fantastic recommendation. I have looked at some of the drugs that are available with IL-1 beta and targeting IL-1 beta, and I have definitely considered that.
Very cool work. Look forward to see where it goes.
Thank you so much. I appreciate it.
Thanks again for your talk. Just a couple of questions. Two, actually. One is, how is notch normally inhibited during ciliogenesis? And did you check your RNA-seq data to see if that inhibition was perturbed in any way during IL-1 beta treatment? Yeah, so notch is usually turned off, like in a healthy epithelium. Notch
inhibiting notch pushes more towards the ciliated cell fate and allows the ciliated cells to recover after the IL-1 beta. It's normally turned off in the healthy epithelium, and there's a good balance of ciliated cells to mucus secretory cells, and that is normal in development. We did look at notch gene transcriptomics before, or like in the same like upregulation or downregulation, and we saw downregulation of some of the notch genes with IL-1 beta and then upregulations of others, but I haven't looked into that pathway as much as I have the ones that I presented here today. We have both notch and Wnt signaling as well that we're also going to consider.
Okay, thank you.
Yeah, no problem. Thank you so much.
It's great data. So I have two questions. First question is, we know the senior cell type depend on different differentiation media. I don't know what differentiation media you used. Do you compare the different differentiation media?
I do, yes. So the first experiment that I did when I joined Esther's lab was a multimedia the effect of the media on differentiation. So we have a very well characterized, published homemade media that we use, and I did a cell culture with that, and we of course pneumocult Pally, the commercially available differentiation media, we tested with that, and then I tested a 1-to-1 or like a 1-to-2, so like or 1-to-1, like half Pally and half our homemade as well. And we ended up, the studies that you see here, we ended up going forward with a half homemade and a half commercial medium, because when we did all of the tests, we found that that was the most like accurate representation of the epithelium. Pally kind of pushes for the overgrowth of ciliated cells. It's a very, very nutrient rich media, and all of the cultures just look like carpets, just ciliated carpets. And then the homemade itself was kind of less rich media, and so it didn't support the differentiation as well. So that's why we did a half and half. It was kind of like a Goldilocks.
Okay. The second question is, we know the IL-1 beta, this dose is related to the CFTR expressions. Did you test the different dose for high dose, low dose? What is the response?
Yeah, so the dose of IL-1 beta that we use in these studies is 10 nanograms per mil. I have not done a dose response curve yet. That is definitely something that is on my radar. However, the physiological concentration of IL-1 beta in CF bowel for lung tissue is very variable. It depends on age, disease severity, infection status, exacerbation status, and things like that. However, the literature shows anywhere from picogram. I'm so sorry. I brutalized y'all's ears with this microphone today. They show like different concentrations from like picograms all the way up to like 40 nanograms per mil. So we picked a kind of modest in between there, but a dose response curve is definitely on my radar.
Okay, thanks.
Hi, Michaela. This is really, really neat work. I just had a question regarding your co-treatment of your DAPT with your IL-1 beta. I'm wondering what happens to the secretory cells when you do that. Are the secretory cells like propped up by IL-1 beta and then reduced by DAPT notch inhibition? Is that what happens, or?
Yes, that is what we see. Yeah.
Okay.
So we see IL-1 beta is in the literature and in our hands as well, in the model that I use, a big inducer of mucus cell hyperplasia and metaplasia. So we see that, and then the DAPT is able to mitigate that as well.
Sure. Okay. Yeah. Thank you.
Yeah, no problem. Thank you.
Great talk. Thank you.
Thank you.
I was wondering about your DAPT experiment and mucus ciliary transport and ciliary bit frequency measurement, if you did that in those DAPT treated cells.
Yes, I have not done that yet. That is definitely a direction that we will go. We're also, of course, we want to look at the functional outputs of the cilia after we we rescue with DAPT treatment to make sure that we're also rescuing function as well. And we're also going to do like ultrastructure, so EM, to look at the actual structure of the cilia to see if we're able to rescue that too. So those are all on the radar as well.
Thank you.
Yeah, thank you so much.
One last question. After chronic IL-1 beta treatment, if you were to remove IL-1 beta from your media, do you anticipate your ciliary differentiation function would return to normal, or would you anticipate some persistent changes in ciliary function?
A washout. I like that idea. Yes. So I anticipate that after you remove the pressure of the IL-1 beta, since these are healthy cultures, that I think it would kind of go somewhere down the middle. I don't think it would be like full restoration to a completely normal epithelium with like the proper ratio of cells and function. But I do think that since these are in a healthy background, that there would be some attempt to form these like normal ciliated cells and restore the ratio of ciliated and mucous secretory cells and the function of the epithelia. Thank you.
Thank you.
Okay, thank you for all the questions. For our last speaker, I'd like to invite Dr. Hiroaki Murano, MD, PhD from the University of North Carolina, Chapel Hill, to finish out the session.
Thank you. My name is Hiroaki Murano from UNC Chapel Hill. I'm a postdoc at Dr. Kenichi Okuda's lab. So our lab is mainly focusing on the studies of biology and physiology of small airways in human lung tissues. I have no disclosures. So, yeah. Small airways are reported to be the primary central part of the CF pathogenesis. And as you can see, usually, and this is the distal lung tissue sections. And when we check the small airways, their airway wall is very thinner and almost same size as corresponding pulmonary arteries. But when we check the CF lung tissues, this is transplanted lung tissues from CF peoples. We can see that most of all of the lung small airways are filled with neutrophil-rich mucus plugs. So we can see that most of the small airways are affected in CF. And generally, human conducting airways are repeatedly divided to make the large airway surface areas. And finally, more than 95% of conducting airways are covered with small airways. So the pathogenetic changes occurring in the small airway in CF should be important. And also, small airways are covered with specific secretory cell types recently detected from the single-cell RNA sequences. This is normal human lung tissues, and these cell types is characterized by the expression of secretoglobin 3A2, SCGB3A2, and SFTPB. And this is RNA scope image of SFTPB highlighted with blue colors, and bronchioles are expressing there is a marked expression of SFTPB, but in the bronchus region, large airway lesions, there are no expression of SFTPB. And importantly, in the Nature paper from Tata Labs, these cell types are highly expressing CFTR. So we are expecting that this cell population should be the very important target of CFTR restoration in the context of CF therapeutic strategies. And also, and these cell types are annotated with different names depending on the articles. Especially they are focusing small airway regions. So some papers mention terminal, terminal bronchiola or respiratory airway secretory cells, but they are almost mentioning the same cell types. So in this study, we call this name, cell types as distal airway secretory cells. And importantly, recent paper mentions that like CF, some muco-obstructive lung diseases shows a decrease or lost in dusk cell types in the COPD or asthma. But in contrast, in non-CF bronchiectasis, non-CFB, the abundance of dusk increased. But so far, there is no reports focusing on the CF small airways. So we, in this study, we aimed that to connect the information of morphometric changes in CF small airway and also what types of changes occurs in small airway epithelial cell layers features, including dusk populations. The experimental strategy is listed here. Thanks to the help of Tissue Core at UNC, we have created the inventory of CF lung tissues, tissue blocks, and also if we have enough amount of tissues, we made frozen tissue stocks as well. From these inventories, we did a morphometric analysis for small airway samples, control for 150 and CF was 370s. And we checked the basement membrane lengths to estimate the airway size and also check the submucosal areas and also corresponding pulmonary artery internal and external diameter sizes. And secondary, we did spatial transcriptomic analysis to elucidate the airway epithelial cell features to compare between control and CF. And also, we estimated that the changes occurring in the CF smaller airway epithelium should be affected by the CF-specific inflammation. To elucidate the cell-to-cell interactions in the CF context, we did multiplex IF imaging analysis on the both control and CF tissues. And finally, to check the epigenetic changes occurring in the epithelial cells layers, we did single nuclei RNA ATAC multiome sequence on those samples. So first result is morphometric analysis part. So as you can see, we can imagine that this CF small airway is dilated if we compare to these airways. But it is because we are selecting the almost matched size of airways. To compare the airway diameters, like CF morphometric studies, we have to reference the original size of airways. In order to do that, we checked pulmonary arterial diameters for reference and estimates the original size of small airways using these pulmonary artery diameters. Of course, after checking there are no differences between pulmonary arterial diameters between control and CF. Then this is the scatter plot of each parameters. X-axis for arterial external diameter and y-axis is bronchiolar diameters, small airway sizes. Then after statistical analysis, we could detect the 0.5 to 2.4 millimeter in corresponding arterial external diameter areas. In those areas, bronchiolar diameter, small airway diameter get enlarged. So using these values, we made subdivided the small airways between dilated lesions and non-dilated lesions. In this case, less than 0.5 millimeter in diameters. Then, using this categorize, we did spatial transcriptomics analysis to see what types of changes occurs in small airway epithelial cell layers. This is a schematic of spatial transcriptomic analysis, and firstly, using the serial section of HE images, so we selected epithelial layers and corresponding submucosal areas and peribronchiolar epithelial cell layers, and if it's available, we also selected the intraluminal mucus plug informations. And after that, we pick up the mRNA and did bulk RNA sequence type of analysis. And this time, today I will show you just only epithelial cell layer information, especially from dilated versus non-dilated areas. Firstly, in dilated region, I mean 0.5 to 2.4 in corresponding arterial diameter sizes areas, we could see the decrease in SFTPB and SCGB382, the marker of DUSCs. And also in contrast, there is a marked increase in the MAC5B and MAC5AC in this area. And interestingly, when we check the non-dilated lesions, there are also, of course, the fold change and p-value are smaller than the dilated areas, but we could see the DUSC marker decrease and also secretory mucin increase in those regions as well. So we estimated that both whole CF small airways shows the tendency that, I mean, DUSC loss, DUSC features loss, and also changes into high secretory mucin producing state in those areas. To confirm this, we did protein staining analysis of these sections. This is the image of the MAC5B and SPB, SCGB3A2 staining on the dilated bronchial lesions, and as detected in the transcriptomic analysis data, we could follow the same trends in the secretory mucins and DUSC marker loss. But interestingly, in non-dilated distal bronchiolar lesions, distal non-dilated small airway lesions, there was MAC5B and MAC5AC increase, but of course there was a trend, but there is statistically no significant differences in the DUSC marker decrease. So from these results, we concluded that mostly in the dilated bronchial lesions, dilated small airway lesions, DUSC marker, DUSC phenotype is lost mainly. Yeah, and also both regions shows a secretory mucin increase. So because usually those secretory mucin increase is occurred by the inflammatory stimuli, so we estimated that some of the inflammatory stimuli cause these phenotypes. So we checked the pathway analysis data from the dilated airway epithelial cell regions. And as expected, we could detect the IL-1 beta related pathway upregulation. Also, interestingly, we also found the type 2 interferon responsive gene upregulations. Type 2 interferon means almost equals to interferon gamma stimulations. Then, when we check the PhenoCycler multiplex IF images, we could see the MPO positive neutrophil enrichment in the intraluminal spaces, and also in the submucosal area, there are massive infiltration of interferon gamma positive cells in this area. To confirm the IL-1 beta and interferon gamma related inflammatory stimuli affects features of small airway epithelial cells. Using, in this case, non-CF derived small airway epithelial cell culture, we treated those cell cultures with IL-1 beta or interferon gammas. After 3 days treatment, we checked the transcripts expression by bulk RNA sequences, and as you can see, SCGB3A2 and SFTPB expression was markedly significantly suppressed in co-stimulation by co-stimulation compared to the single stimulation of the IL-1 beta or interferon gamma. So maybe there is additive effects of both cytokine stimulations. Further understand the CF small airway specific inflammatory environment using multiplex immunofluorescence image phenocycler images. We did neighborhood analysis. Neighborhood analysis is one of the tools to estimate the specific cell-to-cell interactions using the annotated cell and cell distances. So from the 4 independent CF and control section data, we could detect the 9 distinct neighborhoods. And when we plot those neighborhoods into the original images, this is control images, and each plot can recapitulate the original airway structures. So, for example, in this case, light green dots, maybe it's hard to see, is reflecting the airway epithelial cell layers, and red dots, the bottom, is vascular regions. And in CF, we could also follow the same trends. I mean, we could detect the epithelial cell layers, but interestingly, intraluminal spaces are filled with neighborhood 0 annotated one, and in the submucosal area, there is a deep green dots surrounding the airways we detected. So this is neighborhood 2. And when we check the each cell proportionate from the neighborhood, we could confirm the neighborhood 0 was mainly consists of neutrophils, and also neighborhood 2 is from the interferon gamma positive cells. So using different approaches, transcriptomic assay and protein analysis, we could see the 2 important pathways, IL-1 beta and interferon gamma, might be important for CF small airway pathogenesis. So finally, to characterize and understand what types of epigenetic changes affecting the dusk loss in the CF small airways, we did single nuclei at ATAC RNA multiome sequences from 4 CF and 4 control donors. And we could see most of all of the cell types usually detected from the lung tissue single-cell RNA sequences. And when we check the secretory cell population, in this Volcan plot, I just showing the secretory cell 15, but we could see the transcription factor activity. I mean, motif accessibility estimated from the ATAC sequence data. Fox family and NKX families, which is needed for the characterization of distal small airway epithelial cell features. Those transcription factor activities are downregulated in these secretory cell types. So we found that these secretory cell types are epigenetically downregulated in cystic CF small airways. So our summary is CF small airways are partially dilated and show the loss of dusk. And inflammatory stimuli, in this case IL-1 beta and interferon gamma, observed in CF is associated with the loss of dusk, and the activities of key dusk transcription factors are downregulated. And so, yeah, if you need more information, please check my poster. Thank you.
We'll take any questions. Beautiful talk. Perhaps I could start by asking you, so do you think those small airways are getting proximalized? And if so, what do you think is driving the proximalization?
I don't have actually, I think depending on the airway regions, the responses should be different. Maybe more proximal, but I mean dilated part get proximalization. But actually, when we check the more distal part, for example, terminal bronchial regions, the dusk signature is still preserved in CF. Maybe inflammatory differences, because dilated airway regions are filled with neutrophilic mucus plugs, so the maybe degree of the inflammatory stimuli might be different, or the response to the progenitor cell types might be different. Yeah.
Hi, and this is Wenjie from University of Iowa. Very beautiful work. I'm wondering when the transition between the secondary cells to goblet cells, the expression of CFTR, is that increased or decreased? Have a look at the CFTR transcript in those non-CF and CF small airways?
You mean in the spatial transcriptome data?
Yes.
Yes. In the most of ROIs, CFTR transcripts itself are upregulated, but this study contains both stop codon mutation. Maybe I'm not sure which transcripts are recognized as CFTR, but if it's detected, most of the CFTRs are upregulated, but it should not be functional. Yeah.
Yeah. Sure. Thank you.
It was a really interesting and expansive talk. And I'm wondering, how generalizable do you think some of these findings are to bronchiectasis and other disease processes? Like if you looked at, for specifically infection or exacerbation status of the patients, right?
Yeah. When we check the submucosal areas, yeah, we could see decrease in the smooth muscle related gene expression levels, but maybe those cell types are degraded because of the inflammation. But I'm not sure. This study's limitation is their massive inflammatory cells are infiltrating the submucosal areas. So maybe this infiltrated inflammatory cell gene signatures skews the bulk sequence data. And also, when I check the serious red staining in the same serial sections images, especially dilated airways in the collagen fibers get fragmentation types change. So inflammation occurs in the submucosal area, affects the airway wall fragilities.
Yeah,
I think.
Do you think you could learn something specifically about CF if you included tissue sections or samples from patients that had bronchiectasis of a different etiology, like? So you can get for example type 2 inflammation from viral infections and stuff like that. But is there some, because right now, I mean, with the advent of highly effective modulators, we're trying to redefine what is CF lung disease. If a patient is healthy enough to present as like an asthmatic, right? Is that a CF? Like, is this CF asthma, or is this asthma, or what is and your approach is so rigorous and expansive that, I mean, I think we can learn a lot about that.
Yeah. Sorry.
That's great. It's a fantastic talk. I had a quick question. You focused really on the more dilated areas, and the Leuven group, or Mika Boone, has shown that often there is a very stenosed small airway just distal to that area of dilation.
Yeah.
And you kind of talked about the airway that was disproportionately enlarged. My question is about maybe the airway that's disproportionately narrowed, or what they call collapse.
Yeah.
Have you looked at any of the phenotype in that context?
Actually, you mean micro-CT related studies? Yeah, we can see the decrease in the small, especially less than 0.5 millimeter diameter areas. So I think still those small airway distal part area is a mixture of the obstructive bronchiolitis like changes. But we are now checking the how proportionate of those pathologic changes now. But after confirming the each pathologies, especially in non-dilated areas, we are planning to compare those in signatures. Yeah.
Great. With that, I'd like to thank the speakers one last time and thank all the audience for such robust questions.